presentation. So today I want to present our work in our research group about RNA binding proteins. So here we're going to show how we use transcriptomic analysis to characterize a protein that is not well known in Trypanosoma cruzi. So to start, I want to clarify some concepts about Chagas disease. What is Chagas disease? Chagas disease is a public health challenge, and it's a disease that is caused by a Trypanosoma cruzi that have a mammalian host and insect vectors that transmit the infection. So what are the different ways of transmission of Chagas disease? So the main way of transmission is by the insects, but we have other transmission like in food, contaminated food by the parasite, also during the gestation, and also by blood transfusion or organ transplants. So what does Trypanosoma cruzi cause this disease? Mainly is because the parasite goes to some cells in the human host, like cardiomyocytes, and then cause the cardiac cardiomyopathy in the patients. So this is a neglected disease, and is not a drug target or treatment for this disease. So the main idea is to characterize or know the biology of the parasite. So we were interested in how this parasite regulates the gene expression. So the classic that we know is the transcriptional regulation, but the other mechanism that is important is the post-transcriptional regulation. So we are interested in the last one, the post-transcriptional regulation. So in post-transcriptional regulation, we know there is RBPs that are RNA binding proteins, and these RNA binding proteins ligates to the 3-UTR or 5-UTR of the messenger RNAs, and the effect is that they regulate the protein synthesis by inhibition or have a degradation increasing the messenger RNA. One of the proteins that we're interested in is the pumilio proteins, that is the PUF proteins. And in the Trypanosoma cruzi, we found that these proteins have a lot of members. So we have around 10 members of these RBPs. So one of these members is PUF3. PUF3 have this motif in that protein ligates in the 3-UTR. And we know that in other models like Saccharomyces cerevisiae, there is a regulation of genes related to mitochondria. So we will wonder if this mechanism also conserved in Trypanosoma cruzi. For that, we start to characterize the protein. So here we have immunofluorescence of the protein. We transfected the protein and see what is the localization. In blue, you see the nucleus. In red, you have the mark of mitochondria, and in green is the protein. So in the upper panel, we have a control that is GFP, and in the lower panel, it is the PUF3 pumilio protein. So in conclusion, we have the protein have a cytosol localization. Then to characterize more the protein, we did knockout of this protein using CRISPR-Cas9. That is, right now, all the figures are going to have parasite in gray, that is the wild type, parasite in red, that is the knockout using CRISPR-Cas, and also in blue that we have the overexpression. So the idea was to have a model that we have overexpression of the gene and the knockout of the gene, and characterize what happened with the potential target genes. With this model, the wild type, knockout, and overexpressing protein, we use RNA-seq. If the protein have effect regulating the half-life of messenger RNAs, we're gonna show a different expression genes. And this is what we found. This is a PCA that shows the transcriptional profile of these three phenotypes, and you realize there is a difference in the expression. The next question was what is the number of genes that I was regulating. And you see that overexpressed genes have more upregulated genes than the knockout genes. So for us, it was interesting to see if these target genes are mitochondrial genes. We use DeepLog, that is a tool that uses machine learning to localize the proteins, if it's cytosolic, in the membrane, or mitochondrial. So we found that for knockout and overexpressed, we have an overrepresentation of mitochondrial genes. Next, we try to figure it out if these mitochondrial genes that have a change in the gene expression have a motif of PUF3 RNA. So we use the FINO motif finder and trying to find if it's an enrichment of this motif in the 3UTR. So for knockout and overexpressed, we found that more than 25% of the genes have this motif. So that means that a lot of these genes are regulated by PUMIL. We didn't know if it's a direct target regulation or indirect target regulation. So then we analyze in specific what are these genes that are regulated in the both phenotypes and also that have the motif if are mitochondrial genes. If you realize then you found that we have proteins related to electron transport chain. So that gives the idea that PUMIL3 in Trypanosoma cruzi also regulates mitochondrial genes. Here, we use this approach. We have our model, we have the bioinformatic analysis, and we come back to do experiments. This is the good thing when you do bioinformatics that give you new ideas. So the idea was that, or the bioinformatics shows to us there is a regulation in mitochondrial genes. So we explore what happened with the mitochondria in Trypanosoma cruzi in these knockout and overexpressed gene phenotypes. And what we found is this is an electron microscopy. In dark, do you see the mitochondria? These parasites they just have one mitochondria. So we quantify the width of the mitochondria, and you see the overexpressed have a change in the morphology, in the shape. Okay, that gives us the idea that there is a change also in function. So then we move to explore if there is a change in the function of the mitochondria. For that, we use bioenergetics experiments, and we quantify the oxygen consumption, the ATP linked to this respiration, and if you realize, and the data show to us, there is also a change in the function. And the change in the function is higher in the overexpressed gene or the overexpression of the protein. Then we also quantify the mitochondrial ROS production because the last results show that it's an uncoupling on respiratory. So the hypothesis was there is an increase in ROS production. As you realize, we have that without the stimulation, we have an increase of ROS in these two phenotypes, the knockout and also in the overexpressed protein. So what we have here in conclusion is that the Trypanosoma cruzi PUMIL3 regulates the expression of target mitochondrial associated genes, highlighting a potential role in energy production, cellular metabolism, and other mitochondrial-related functions. Also, this gives us the idea that this is a potential protein to target for the disease. If you target this protein in the parasite, we could have an effect on the treatment of the disease. So these results will have published this year. If there is a publication, if you have interest. And I will also acknowledge our team, Ana Maria, that is the PI, Omar Triana, that is the director of the group, and all the people that was involved, like Jader, Ana, Diana, and Luis from Equity University that helps us in this project. Also the financial by the MinCiencias and all the grants from the university. Thank you. Yeah.
You showed us a PCA
Yeah.
with 3 conditions. This was done with the expression of all the genes?
No, the different cell express genes. Yeah. We use DE genes for construct this PCA. Yeah.
But you have expression from all the genes?
Yeah, we have expression from all the genes.
Because my question was, did you try maybe to construct a co-expression network to see if there are relationships of the gene coding for this protein with the gene that could maybe give the idea of the role of the protein?
Yeah, the co-expression we didn't try, but this is a nice idea. But what we did is that we have the 2 phenotypes, the knockout phenotype and the overexpress. We was thinking that was an overlap of different expression genes in these 2 phenotypes that we didn't found. So one thing with this is that we didn't roll out that it's a big family, and maybe the motif is shared between other families of the medial proteins. We knock out just PUF3, but the parasites also have the other ones. So this is the explanation that we explained that this overlap is not too high. Okay, this is one of our limitations. Yeah, because try to work with this huge family that share the motif. So you knock out one gene, but the parasite have other members of the family. So the ideal is to have multiple knockouts of these genes, where it's experimentally so difficult. There is another 2 questions.
Thank you, Geysson, for your presentation. It was a talk. I'm not very acknowledged, or I do not understand this model completely, but I was wondering, why did you choose the 3' UTR to look for the motif and not the 5' UTR?
Yeah.
Is that related? Because I see that there are also other ribosome binding proteins.
Yeah.
In this side.
Yeah. The main reason is because we also have information for Pumilio 3 in Saccharomyces cerevisiae. So in this model we know that the motif is in the 3 UTR, is mainly in this region. But you are right, maybe it's the mainly but not the unique one. So yeah, we didn't analyze the 5' UTR. It's a good idea, but the main reason is because previously it was reported that is in the 3 UTR the most enriched region that we have this motif.
Would you use an genetic approach to get those regulatory functions of that?
Yeah, flat him after the